At least the introduction part, etc.
Yes.
Dr. Chauhan, is that okay with you?
Yes, sir.
Excellent. Good evening, everybody, and welcome back to a new edition of the D.Y. Patil Postgraduate Psychiatry Knowledge Series. We have with us our dear friend and one of the most prominent academicians of our times, Dr. Malay Dave, to speak to us today about the humble mitochondrion. I will have Dr. Chauhan introduce Dr. Malay Dave in a bit more detail, but it gives me great pleasure to invite both of you with us today. And Malay has been a dear friend, a senior colleague for many years. We've learned a lot from him in all these years, but it's always a pleasure to listen to him back again because he's always got something very interesting up his sleeve, which leaves a little gobsmacked, in terms of some new learning. We are very happy to have with us Brigadier Dr. V.S. Chauhan. He is a professor and head of Department of Psychiatry at the AFMC Pune. He's done his MD Psychiatry from AFMC as well. He's been teaching undergraduate and postgraduate students for close to 2 decades now. He's received several military awards. He's received the BDE General Commendation of the UN Mission in 2008. He's received the COAS Commendation in 2011. He's got several academic achievements to his credit as well. He's received the Colonel Kripal Singh Award for the best paper in psychiatry in 2010, 2012, 2019. He's received the Best Armed Force Research Project Award in 2021. He's published several papers in the Indian Psychiatric Society, as well as he is a member of the Advisory Committee on Mental Health Strategy for UN Troops. He is an examiner at various forums, the MUHS, the National Board of Examination, as well as the WB UHS. And we are really glad to have him with us today. Thank you, Dr. Chauhan, for making the time and being a part of this venture of ours. And we invite you to take over the proceedings. I'll come back at the end of the program to deliver the vote of thanks. Over to you, Dr. Chauhan.
Thank you, sir, for the nice for the introduction, and it gives me great pleasure to host this meeting. I thank D.Y. Patil as well as Dr. Ashwin Kalra to have given me the opportunity. Am I audible?
Yes.
Yeah. To have given me opportunity to moderate this session. The speaker for today is Dr. Malay Dave, sir. He needs no introduction in this forum, but it is customary that I should talk about him a little. He's a consultant psychiatrist and is a private practice. He's got 12 years of PG teaching experience, and he's received multiple awards, which includes Bombay Psychiatric Society Silver Jubilee Prize, Dr. Anil V. Shah Best Paper Award. Got a number of publications. He's been honorary secretary of Bombay Psychiatric Society in 2016-17. He's a member of, he was a member of IBS UG Education Subcommittee, and he's an advisor, currently an advisor of IPS Biological Psychiatry. His area of interest is neuroscience and genetics, and today he's going to talk about the humble mitochondria. As you all know that as the generator of metabolism, mitochondria are the unifying link for the known risk factors for many of the mental disturbances, which includes genes, hormones, neurotransmitters, adverse childhood experiences, rejection, loneliness, and it is very highly sensitive organ or mitochondria, which reacts to both internal and external cues and external environment, and it is readily adapts to that. In the past few years, there has been a few research in the areas of mitochondrial dysfunctions, especially in stress-related disorders, anxiety, depression, schizophrenia, bipolar disorders, and autism spectrum disorder. So without further ado, I would request Dr. Malay Dave, sir, to take on the proceedings, sir.
Thank you, Dr. Chauhan, for the kind words. Thank you, Rashmin, for inviting me once again to speak on this forum. I'll share my presentation and then we'll start. Yeah, is it visible?
No, sir.
Is the PPT visible?
No.
Because I've shared the screen.
You can see your screen, Malay, but not the PPT per se. I think you have to open that.
Yeah, is it now? Is it
No,
seen
no,
now?
no. We cannot see your PPT yet.
Okay, just one sec. Let me open it again. Visible?
No, unfortunately.
Not yet, sir.
Because it is telling me that viewers can see this screen. So, seen anything?
No, Malay.
No? Just a second. You can see the sort of, no?
You can see your screen, but not the PPT. I don't know. The PPT isn't opening, I guess.
Wait. Can you see this first slide? This one?
No, Malay. We are just seeing your folder open. Where you have
Can you see this one? You can see the folder. Okay. Okay, just 1 second. Let me see what's going on. I don't know why this is happening. Okay, let us see. Can you see it now?
Yes.
Okay, check, good. So, and am I audible also as well?
Yes.
Yes, sir.
Yes, you are.
Okay. So, thank you. And sorry for that glitch. There was something wrong in the way that I was opening. So, I'll be talking about the mitochondrion today. As Dr. Chauhan pointed out that it is a new area of research, not only for psychiatry per se, but for the entire branch of medicine, all the fields of medicine, all the branches of medicine, wherein we treat patients, see patients, diagnose them and treat. And more importantly, we need to know something about the mitochondrion because it is a kind of a universal factor in the causation of many non-communicable diseases, psychiatric disorders being a part of that. So we'll start. So what is the mitochondrion? Basically, it is something like this, which is seen in a eukaryotic cell. So it is a double membrane bound cell organelle, which is seen in eukaryotes. Double membrane means it has got two membranes surrounding which encapsulates the whole thing, and it's seen in eukaryotes. Eukaryotes are human beings and other mammals who have got cells which have a nucleus which is surrounded by a nuclear membrane. This is the definition of a eukaryote. So basically all mammalian cells. The mitochondria are associated with ATP generation, and therefore they have been called as the powerhouse of the cell, because ATP is the energy currency for all processes that happen inside the human body, anywhere in the human body. The term comes from two Greek words, mitos meaning thread, and chondrion meaning granule or grain-like. It was first called as bioblast when Richard Allman discovered mitochondria in the 1890s. And Carl Benda coined the term mitochondrion in 1898. So it is almost 100 years and 140 years plus for us that we have known mitochondria in one form or the other. Mitochondria are efficient, adaptable, bioenergetic machines which have a functional and compositional diversity. So some of these things we'll see in some detail as we move. What we need to understand is that the mitochondrion was not a part of the cell, not a part of the eukaryotic cell millions and millions and billions of years ago. Basically, mitochondria were bacteria. These were alpha proteobacteria. Somehow this alpha proteobacterium encountered a eukaryotic cell. The eukaryotic cell kind of ingested this bacterium, and then they decided that no, the cell decided that I'm not going to digest this mitochondrion. And the mitochondrion said, and that bacterium said that, okay, thank you for not eating me up, I'll help you. And since then, since those few billion years ago, they have this endosymbiotic relationships. An endosymbiotic relationship means that there is an organism within an organism, and both live in perfect harmony, helping each other out. Mitochondria are one of the only two endomembrane systems, the second being the endoplasmic reticulum. So there are two systems that psychiatrists need to know. One is the mitochondrion, which we are discussing today. Maybe some other time we'll talk about the endoplasmic reticulum, which is also a double membrane structure. And not only are these two systems, that is the mitochondrion and the endoplasmic reticulum, kind of distinct or apart, but they have direct and intimate interactions between themselves. Also, mitochondrion, even though they appear in diagrams like this, what we saw, like orange structures far and wide spread out, it is not like that. It will form a network which spans throughout the cytosol. So in a cell, depending on situations, there would be multiple mitochondria which are kind of connected to each other in many ways. We'll see that. So the easiest way is this old image where we understand that mitochondria is something like this. It looks more like a kind of a bread which has got all these striations, but this is an electromicrograph. It's 200 nanometers this much, so they are pretty small. A three-dimensional view would appear something like this, where this is the mitochondrion, these are the membranes, this is the internal structure, and this yellow thing is the endoplasmic reticulum. So this is only a mitochondrion. This is a mitochondrion with endoplasmic reticulum, which is kind of wound around it. And here, if you look at the C diagram, C picture, you have got one pink round, another pink round. These are two distinct mitochondria coming together. So what is the structure? What is the anatomy, or let's say the microanatomy, or what do we know about the mitochondria as an entity? So this is a more kind of a diagrammatic view. And what does this view tell us? That this is something like a bean-shaped structure. This has got an outer membrane. This has got an inner membrane. There is an intermembrane space. And this inner membrane is like folded, which form the crista, so to say, cristae. And then there is this matrix, which is the space within the bound by the inner membrane. And then there are a lot of these things inside. There is some DNA inside. There are ribosomes. Then there are these kind of button-like things, which are the enzyme system. So this is roughly what is seen in the mitochondrion. So a mitochondrion has two distinct membranes, the outer and the inner membranes. And it has got two functionally diverse compartments, the intermembrane space. This intermembrane space is between the outer and the inner membrane. So to see it once again, this is the intermembrane space, outer membrane, inner membrane, and the space between, and the matrix, which is the second space, which is the space contained within the inner membrane. So this outer membrane is a smooth lipid enriched surface. It is made up of very old kind of proteins, which are called as ancestral beta barrel proteins. It has got certain kind of channels, which are called as VDACs, voltage dependent anion selective channels, or porins for short. And they allow molecules up to 10 kilodalton in size, so very small. So this surface has got pores, which are these VDACs or porins, and they allow very small molecules lesser than 10 kilodaltons in size to enter. Also are present alpha helical membrane proteins. And these proteins are certain proteins which have got one transmembrane domain, which are called the MFF and MIRO. Now, what are these proteins useful for? One is the older proteins, one is the alpha helical proteins like this. These are the receptors for the mitochondrial division dynamics and motility. So how mitochondria are going to come to close to each other, how they are going to communicate with each other, and like we saw in this diagram here, how a mitochondria is going to interact with the endoplasmic reticulum, here how two mitochondria are going to talk to each other. This is going to be decided by this alpha helical membrane proteins MFF and MIRO. So a very detailed kind of structural, pictorial, biochemical, anatochemical view of the outer membrane is this. So this is the outer membrane proteins. This is a cytosol. This is the inner membrane. This is the intermembrane space, and this is the matrix. So all these proteins are there on the surface. This MIM is single, single domain. These are the alpha helical proteins, all these semi-rounded ones, right? And they basically will kind of give the mitochondria the ability to do a lot of things, that is intercommunication and things like that. And this is the inner membrane. Now this intermembrane space, again, this intermembrane space between internal and external is a very small compartment, but it contains 5% of all the mitochondrial proteins. And what do these proteins do or help in? They help in protein and lipid biogenesis, metal homeostasis, redox regulation and oxidative folding, apoptosis, and mitochondrial dynamics and structure. This inner membrane is a protein-dense membrane. So a lot of proteins, lot of receptors. It is highly impermeable. So it doesn't have all those pores like porins or VDACs. And it is folded. It is not straight. It is folded. So it is folded like this, because it is not porous and it has got a lot of protein. So if it was just a single thing, it would get overcrowded. It will not have enough space. So it is folded, folded and folded and folded. So these are the cristae, right? And what is the function of this? Now this internal membrane has got the electron transport chain, also called the respiratory chain, with the respiratory complexes, which are complex 1, 2, 3, 4, and a turbine-like ATP synthase, which is 5. So 5 complexes, right, that drive the formation of the cristae. Means they will decide how active the cristae are and how many folds are going to be there. More folded it is, it means there is more surface area available for more ATP production. So complex 1, complex 2, complex 3, complex 4, and complex 5. This is what is there on the inner membrane, diagrammatically like this and something like this. So this is what we saw, these dots. These are those systems. What is the matrix? Matrix is the space inside. It contains mitochondrial genetics. Biosynthetic reactions happen there. The most important of them being the TCA cycle, or tricarboxylic acid cycle, which basically oxidizes carbon backbones of nutrients, NADH, FADH2, reducing equivalents, and these are used by this system, respiratory chain system, to produce energy or ATP. So what is this proteome? Proteome basically is the whole lot of proteins that are present in the mitochondria, both in the intermembrane space, localized to the intermembrane and the matrix, and they are important for various aspects of metabolism and functioning. But it also has got over 200 species of lipids inside. And what are these lipids? Glycerolipids, phosphatidylcholine, ethanolamine, cardiolipin, which is very specific to the mitochondria, and these have got bacterial origins. Remember, as I mentioned earlier, the mitochondria are not were not a part of the cell before. They are bacteria which have been ingested, and kind of wiped clean of certain problems, things, and now remaining there in the cell and doing a lot of function. But by character, it is a bacterium. Okay, so once you have got that straight, so this is the cell membrane, this is outside of the cell. The mitochondrion, we have seen there's an external membrane, internal membranes, crista, intermembrane space, matrix, and this diagram only shows one aspect of the mitochondrion, that is the TCA cycle, which utilizes pyruvate from glucose, which has entered into the cell. And then there is stage-wise oxidative phosphorylation, progressive electron loss, and finally we have got ADP, which unites with an inorganic phosphate, and we get ATP, which is the currency, energy currency. Now, what is important is, now that was the basic structure that we have learned in, let's say, first MBBS. This is what the mitochondrion is. But the important thing is what does it do, how things are done, and what drives the mitochondria. So first of all, let's have a little bit of focus on the genetics. So as I mentioned, after deciding to stay in the eukaryotic cell, this bacterium, the alpha proteobacterium, lost most of its genome. So most of the cells, most of the genome was lost because it was problematic. So they decided, okay, you stay here, but we'll do away with this. And the remaining genes, many of these remaining genes were transferred to the nucleus of the cell. So any cell in our body, in the human body, has got its own genes, uniquely human genes or unique mammalian genes, plus a number of genes from the ancestral genome. So the mitochondria is there in the cell, and it has donated a substantial part of its genome to the human genome. So current eukaryotic genomes have more than 600 genes of those bacterium. Now, if we have got plenty of genes, some say that we have got about 20,000 plus genes in the human cells, of which more than 600 are of the alpha proteobacterial origin, and donated by whom? By the bacterium which became a mitochondrion. And 99% of the mitochondrial proteins are encoded by the nuclear genome. Means mitochondrion as a structure, we saw that there are so many things that the mitochondrion has: membranes, which is lipids, different kinds of lipids, proteins. Then there is then all those enzymes that are needed for the various reactions. So all of that, 99% of the mitochondrial proteins are encoded by the genes that are present in the mammalian nucleus. So there are about 2000 nuclear DNA coded genes, which decide what the mitochondrion is going to look like, what it's going to function like, and thousands of copies of the maternally inherited mitochondrial DNA. So the mitochondria have got two genomes. One is the genes that it donated billions of years ago and now are a part of the human genome. And then there are certain DNA elements which are called as strictly mitochondrial DNA, which are present in the mitochondrion. So if we want another look, this is what it is. These purple structures, these are the mitochondrial DNA. This DNA is separate from the DNA of the cell, which is there in the nucleus. So a cell will have two genomes. One is what is there in the nucleus, and second, what is there in the mitochondria. And mitochondrial structure and function is decided by those 600 genes which are there in the nucleus of the cell, plus this mitochondrial DNA. So the nuclear DNA, what do they do? They are responsible for the anatomical components of the mitochondrion, for mitochondrion intermediate metabolism, mitochondrial biogenesis, and regulation of the mitochondrion in an overview. Whereas the mitochondrial DNA, those ring-like things, are critical genes for oxidative phosphorylation and for the expression of genes which do all oxidative phosphorylation activity. So in short, all the energy production, all the ATP production, and all the crucial reactions which are needed to produce energy and how this energy is utilized are the mitochondrial DNA. This gives the structure and this gives the function. So this mitochondrial DNA is very small, 16.6 kilo base data, which codes for 13 proteins only. 12S and 6S ribosomal RNA, 22 transfer RNAs for mitochondrial protein synthesis. So this is very small, 13 proteins, and these are the 2 ribosomal RNAs and these are the transfer RNAs. But what does this mean? This means that this is responsible for only a very select transcription and translation of proteins. And because there are very few proteins involved, anything that can go wrong with even one of that will have a dramatic amplification in cells. If there are thousands of proteins or thousands of genes and one is not functioning well, the effect may not be that great. But you have only 13 proteins. We have a very small mitochondrial DNA. So even a little bit of a problem and things can go completely haywire. And what does this give us? What is the advantage of having a mitochondrial DNA? So because mitochondrial DNA is specifically related to energy production, because it doesn't have any other activity, transcription and protein translation, because this is something which goes on all the time, this consumes the highest amount of cellular ATP. So this process became very easy, very efficient, because energy is always available. And eukaryotic cells, because something like this was happening all the time, because we have a lot of how would you say, no shortage of energy, we have a surplus of energy, eukaryotic cells could actually evolve into relatively large and functionally diverse entities. So eukaryotic cell was not only one cell, but as it evolved gradually, it started forming specialized cells. These specialized cells started organizing into various small organ systems. And so we have different kinds of, let's say in the human body, we have got hepatocytes, we have got renocytes, we have got cardiomyocytes, we have got neurons, and different kinds of functionally diverse entities. So the loss of the ancestral genome is responsible for evolution of new pathways for mitochondrial and lipid biogenesis. So not only did the eukaryotic cell prosper, evolved, specialized, it also streamlined the mitochondrial process, because this only deals with energy production. So it keeps on getting refined. And so mitochondrial protein and lipid biogenesis also became very, very strong. Now, mitochondrial gene expression levels vary significantly in humans, not from human to human, but even in individual tissue and individual cells. The mitochondrial DNA variants, okay, what do they contain? Now, what also we need to understand is that there are different mitochondrial DNA in each mitochondria and in each cell. So they contain a lot of variants. And what are these variants? These are ancient adaptive polymorphisms. So again, mitochondria billions of years old. So even their genes, even the genes that have been transmitted over the generations, they kept on thriving well because of polymorphisms. Really recent deleterious mutations. So as the genome progressed over millennia, centuries, decades, it started accumulating mutations and developmental somatic mutations. So whatever was there started millions of years ago, those polymorphisms, deleterious mutations that have been accumulated over time, and developmental somatic mutations is as the development progresses, as we become older till we die, those mutations, somatic mutations. Now, this the mitochondrial DNA is maternally inherited. Why it is maternally inherited will come a little bit later on. So the male and female maternal DNAs do not mix and this cannot recombine. So the mitochondrial DNA is maternally inherited. The nuclear DNA will have a mix. This is strictly, so we saw that there are two DNAs, the one in the nucleus and the one in the mitochondria. This will get transmitted like any other cell. The mitochondrial DNA goes only from mother to child. Son will not transfer that, the daughter will. So a single mitochondrial DNA is called as a haplotype. And a cluster of related mitochondrial DNA haplotypes is a haplogroup. So again, for example, if you look here, single one is a haplotype. This becomes a haplogroup. Now, mitochondrial DNA haplogroup, there are lineages. And these are geographically delineated and they are found in indigenous populations. There is a diagram which I will show you, which will make things a little bit easy. And how has this helped? It has helped in the adaptation to new environmental factors such as alternative food resources, activity demands, altitude, temperatures, and diverse infections. And so they become unique to a population and to a person in that population. So it is something like this. Millions of years ago, there were only eukaryotic cells, and eukaryotic cells were found and they evolved into human cells in Africa, let's say some million years ago. So that initial human concentration in Africa, for example, would have a certain haplogroup of mitochondrial DNA. But then what happened? People started moving. People moved away from Africa. They moved into, let's say, Europe. Then they moved into Asia. Then they moved into North America, South America, Australia. And as they moved, they encountered various geographies. And geography is not only a place, but it is an environment. It's an environment which has got an altitude. It has got water availability, oxygen availability, plant availability, different kinds of microorganisms, viral, bacterial. So this mitochondrial DNA, which originated here, kept on changing. So all this that we see, N, IWX, YA, S, and all this. So this numbers are how how many years ago where we were. So let's say 200,000 years or 2 lakh years ago, we were in Africa. Then we went to North Africa, 65 to 70,000. Then Europe. Then again, from there, we moved on to Asia and so on and so forth. So not only did people move, the mitochondrial DNA also moved. It covered the entire globe. So what we are going to have is people all over the place with the same or a similar genome, but the mitochondrial DNA will keep on changing from place to place because of the difference in place to place. And that's why it is said that mitochondrial DNA lineages. So this is a lineage. So there will be one lineage here. L0 is the original one, lineage 1, lineage 2, lineage 3. Then it moved out. So different lineages. So these are geographically delineated indigenous populations. The populations that, let's say, for example, stayed in Africa will have one lineage. And the one which came to Europe will have another lineage. The one we moved out into Asia will have another lineage. And so this is what has happened, geographically delineated indigenous populations. Why am I stressing on this is because when we look at disease states, we don't need to look far. I'm talking not in the sense of other physical illnesses, but in terms of even psychiatric illnesses, degenerative disorders, what happens is that many a times when we look at the genome, let's say of people in Asia who have got Alzheimer's disease, we are going to find that some ancient DNA is present more in Alzheimer's disease patients or more at risk patients compared to those who do not. So this ancient DNA, if it comes in circulation, it will be in circulation because it is maternally transmitted. It has not been wiped out. Some of it may come, not the 100%, but even 2%, 5%. This can increase the vulnerability to illnesses. If we think about what happened in COVID, that was the time when people had the time, opportunity, and the means to study viral infections in different populations. So a lot of people of the Caucasian group, the whites, they died. Whereas the same age group population, the Asians, had some problems, but the mortality was less. So it was found that certain ancestral DNA concentrations were higher in those people who succumbed to their illnesses and not present in those Asian ones. So they got infected, but they didn't have a bad outcome. So this ancient mitochondrial DNA has this kind of a problem. Now, mitochondria are related to bacteria, that is clear. So mitochondrial DNA replication and transcription enzymes originate from bacteriophages. So what happens is that this phage polymerases, let's say like polymerase G or polymerase gamma. What do they do? They maintain DNA, they repair DNA. So when we know that bacteriophages, means the viruses, when they attack, when they get into a bacteriophage, they are going to they'll be stripped out of everything. Their DNA will get integrated in the DNA of the bacterium, and then they will multiply there. But these viruses, for their DNA to get incorporated into the host DNA, they require enzymes which will first allow that insertion and second allow the multiplication. So they are phage polymerases. They are high processing, maintain the high mitochondrial DNA copy number. Now this mitochondrial DNA copy number per cell varies according to the cell type and is highly regulated during cell fate transitions. Mitochondrial DNA is maternal lineage. So what happens to the paternal mitochondria? Paternal mitochondria are generally found in the sperm tail. So when fertilization takes place, the tail goes away. So the part of the sperm which enters the oocyte or the ovum has next to nothing in mitochondria. Also, during the process of spermatoid sperm maturation, the nuclear localization of a spermatoid cell, specific form of this is mitochondrial transferase type A, this enzyme gets destabilized. So the mitochondria are not there. The human oocyte, on the other hand, has got about 200,000 copies of mitochondrial DNA distributed after fertilization during division, right? And the replication of this mitochondrial DNA assumes only after the blastocyst stage, so means after a few cell stage. So in the initial few cells, these copies get distributed, and then there is going to be cell division, and then this will get replicated. So this kind of mitochondrial DNA gets transmitted to a generation without much of change. So during oocyte development, there is a mitochondrial DNA bottleneck which selects against pathogenic variants, especially protein coding variants of mitochondrial DNA. This has become a focus for intervention with regards to how to prevent mitochondrial DNA transmitted diseases. So this is something like that. This is a representation of the mitochondrial DNA. It is a ring-like structure. This is how it starts the initiation, copy, this separate, and so on and so forth. So what are the mechanisms? What do the mitochondria do? So what we have talked about now is the anatomy, the genetics, and what do the mitochondria do? First and foremost, it does produce ATP. So this is the electron transport chain. We saw that in the previous diagram. There are 4 complexes and 5th one. So NADH, FADH, reduction. So at each and every stage, there is an electron transfer from C1 to C2, 3, 4. Okay. And whatever is available is used by ATP synthesis. This is the complex 5. And we have got ATP. So this is basically the reaction. So what does it do? Energy is there, but more important is setting up a metabolic program. Means providing the necessary energy and molecular precursors for various cell functions based on nutrition, stress, and differentiation status. Now, these respiratory complexes we have seen here that they are separate 1, 2, 3, 4, but they can also work together to increase the efficiency of respiration. So, RC1, complex 1 and 2 can combine together. And electron transfer by this complexes is coupled to proton transport. So O minus H plus, and this difference across the membrane is around 150 to 200 millivolts across the internal membrane, and this powers the fifth complex, which is ATP synthase, complex 5, right? And this produces a gradient. This gradient is also important for a lot of things. What? Mitochondrial fusion, protein import. It is a sentinel for mitochondrial health. So if the energy level is low, the whole metabolism will shift. There will be stress responses that can start. There can be mitophagy. Mitophagy is, okay, this mitochondrion is overworking, not functioning well. It has to be removed for circulation. If it is too much, it can lead to cell death. So what is this potential? How much of it is maintained? The cell will decide enough. Okay, fine. Otherwise, all these other processes will come into play. So mitochondrial health. So without going into too much of detail, we have seen enough of structure, function. And so these are all the possible things that happen in a mitochondrion. So we start with, let's say, ATP. Okay. So energy. But then there is also redox metabolism, redox regulation, nutrient metabolism, amino acid metabolites, so growth, repair, sulfur metabolism, nitric oxide and carbon dioxide, so vascular tone, reactive oxygen species. So reactive oxygen species, basically electrons, more of them, but that is a signal for stem cell differentiation. Lot of it, more of it can create a problem. So copper and iron clusters, innate immunity, lipids, so cell membranes, peptides, cofactors, that is heme, calcium regulation, any function, any metabolic process which is important for the cell to survive, everything is done in the mitochondria, or mitochondria provide certain molecules or precursors for those reactions. So mitochondria, but it helps in the glucose cycle, which goes on in the cytoplasm, the tetrahydromethyl folate to formate that cycle, glutathione, homocysteine, purine synthesis, everything is modulated inside the mitochondrion or with the help of mitochondrion in the cytoplasm. Mitochondrial shape and distribution. So we saw that, you know, whatever diagram you see, mitochondrion will be seen as a kind of a kidney, small kidney, but it varies. It varies dependent on mitochondrial division, fusion, and motility. We'll see that in a bit. Also on the embryonic stem cells. So when cells are developing, mitochondria are fragmented and underdeveloped. But as the cells become more efficient, the structure changes. Also, different function in different types of cells. So there are different mitotypes. So in brown fat cells, where mitochondria will contact lipid droplets, because brown fat is highly metabolic compared to white fat or white lipid, which doesn't oxidize so well. So here, the mitochondria will have a different function, specific lipid processing, and it's a different mitotype. So this is something which happens at different points in cells. So stem cells, they have got different kind of activity, that is oxidative. Proliferative cells, so cells are dividing, so anabolic but glycolytic, so here glucose comes in. Differentiating, so again oxidative, so different aspects of cell growth, cell division, different kinds of energy requirement, and this metabolic map or metabolic framework is set by the mitochondria. Now, not only that, if you look at this thing, so first is starvation. Again, there will be a different kind of a pathway. If we look at thymus and immunity, another kind of a pathway. This stem cells and mesenchymal stem cells we saw. This is something more about mitophagy. DNA damage means aging, apoptosis, whatever be the type of the cell, the type of the function, and what is needed. If you see the mitochondria, different shape, more of tubular, rounded, okay. Here more of apoptosis, so undergoing degeneration and things like that. Okay, this is where if we see, I talked about that brown fat kind of thing. So here we have a mitochondrion. Now it is very close to the endoplasmic reticulum, and together they are going to process this lipid droplet. So again, it's not only the shape, size, but how closely it works with the other cell organelles like endoplasmic reticulum. Again, a more detailed diagram. How does it manage its own self? It's a separate, we say it's an organelle part of a cell, but it is a separate entity by itself. So two main things, fission and fusion. Fission is breaking, fusion is coming together. So the primary fusion factors are, this is a fission. So break one cell into two, or fusion, that is two mitochondria joining to form one. So there are again a lot of factors. DRP1, again, so many factors managing DRP1, and final fission, MFF, and fusion, MF1 and MF2. So fusion. So basically the primary fusion factors are OPA1, MFN and MFN2, which bind inner and outer membranes. So mitochondria 2 will join together and work better. Fission is mainly mediated by DRP1. Okay, we saw that ring-like thing here, DRP1. Okay, and here fusion is MFN. So what happens is, when we want more activity, when the mitochondria decide that let us combine our strength, there is fusion. When it is not needed, it might break down into smaller ones, a bit of fragmentation. But if the wear and tear is too much, then there is mitophagy, means that mitochondrium is going to be broken down, that is lysed by the lysosome. Okay, so lysosome will happen and that will be taken out of circulation. Another thing is nuclear encoded mitochondrial protein biogenesis. So we are talking of how the mitochondria regulate itself. So one is by joining with other mitochondria, or breaking down into more mitochondria, or getting out of circulation. Transcription wise, transcriptional control is peroxisome proliferator activated R gamma, PPRGA co-activator, this one. This PPRGA will come to it in a few minutes. Translational control and post-translational control. Plus, of course, there is a lot of lipid biogenesis, lipid transport, contact sites. So all this will manage how the mitochondria modulates itself. Now, whenever there is a mitochondrial dysfunction, the things go in a reverse way. Okay, because if we go from bottom to top, it requires more energy. Something is going wrong. It is going to move a little bit behind on those steps. So there will be remodeling of metabolic pathways, which is more now guided to, means more going to focus on repair removal rather than managing other metabolism. But when the capacity is exceeded, then the cells will die because of apoptosis, ferroptosis, pyroptosis. And mitochondrial proteases. So these are the first line responders for mitochondrial quality control. Okay, so the mitochondria will, kind of get broken a part of it or the whole of it. Protein signaling, this is where the proteases will come into play. Mitochondria also deal with innate immunity and inflammation. So tissue damage, mitochondrial antigen release. Viral infection will release the mitochondria contents into the cytoplasm. Activation of cellular pattern recognition receptors and downstream cascade of innate immunity response. Means an innate immune or inflammatory process can start within the mitochondria itself, which will also lead to upregulation of nuclear transcription proteins, basically starting off a cytokine response, mitophagy, because mitochondrium is too active, let us remove it. And this inflammation, if it is beyond a certain point, will drive pathogenesis. And there are certain mitokines. Now, all these chemicals, pro-inflammatory, anti-inflammatory, chemokines, these chemicals, they will manage the inflammation level. But by itself, the mitochondria also have certain chemicals called as the mitokines, okay, which are important for managing the inflammatory state and communication between the mitochondria. As we age, mitochondrial functions also change with aging in a tissue-specific manner. There is focus, shift towards mitochondrial repair function at the cost of ATP. So now less energy is generated and processes are going towards repair, because as we grow older, cells undergo more and wear and tear, more tear. So they have to be replaced. So more is the focus on that. This leads to changes in transcription and translation. And if it goes beyond a certain point, now we have understood that this contributes to most age-related degenerative diseases, whether it's dementias, whether it is Parkinson's, whether it is anything, this is at play. And this altered metabolism can also cause adverse drug effects. So as we age, there is a slight shift in how the mitochondria function. And that is where we are going to find that, okay, previously this was not happening to me, I could tolerate this. Now we get a side effect with the same medication. So why do we need to look at this as psychiatry is one? We do not have one pathophysiological explanation for psychiatric disorders, no coherent, different ones. And we assume that symptoms like mood symptoms, memory symptoms, since they come from the brain is the primary seat of the problem. But what we need to understand is this: the brain is only 2 to 3% of the body weight, but it utilizes 20% of all ATP. So it is very, very energy consuming. What is the first most energy consuming? All the DNA processes. The DNA gets translated, the DNA gets transcribed, that process, because that is all the time. And the second one is the brain. So what happens is, even if there is a mild problem with the mitochondria, mild bioenergetic effect, brain gets affected first. So mitochondria, the bioenergetics of the mitochondria will decide how a certain organ functions. And in that, organs which require the most energy will have the most negative effect if even slight changes are possible, slight changes happen. Mitochondrial disease is present at any age, in any organ system, in any tissue or multisystem, and can have any inheritance pattern. So there is nothing fixed about it because there are too many things at play. Even if you look at genetics, there are two things: the nuclear DNA and the mitochondrial DNA. One, mitochondrial DNA, maternal inheritance, and the other can follow any kind of a thing. Also, the mitochondrial DNA has a very high mutation rate. So a lot of changes can happen. So with the mutations, we have mixed things, some normal mitochondrial DNA, some mutant DNA, which is called as heteroplasmy. And because heteroplasmy again is not one thing, it has a gradient. We have got graded defects and variable phenotypes. And because of so many genes, they will have an effect finally on oxidative phosphorylation and bioenergetics. Now, the other tissues apart from the DNA and the brain are heart, kidney, muscle, and endocrine systems, which require high energy. So even mild mitochondrial dysfunction will not have an effect only on the brain, but have an effect, let's say, on endocrine and metabolism. So we have diabetes and metabolic syndrome, which get associated with psychiatric diseases. Now, ancient DNA, mitochondrial DNA also contributes, those haplogroups which I mentioned about. And heteroplasmy in this kind of thing, again, if it is 20 to 30%, we have one presentation like T2DM or autism. If it becomes more, we have got multi-system involvement. If it is 100%, we have got a different kind of a problem. Mitochondrial DNA mutations accumulate with age and they can be progressive. And also, as I mentioned, these were originally bacteria. So when viruses get affected, or when there's a viral infection, this will get activated first. And when they get activated first, they actually expose themselves to viral DNA. And that is why wherever there are a lot of mitochondrial activity in whichever cell, if there's a viral infection, the risk of those viruses having a serious effect is more, because the viral DNA can get easily or easier get integrated into the host DNA. So this is what actually happens in CNS viral infections. Because again, neurons and glia are very active, lot of mitochondrial DNA, lot of mitochondria. And so it's kind of a passport for viruses to get entry into the host DNA in the CNS. So apart from this, what are the ways in which other things can happen? So mutations we have talked about, aging, reactive oxygen species, sedentary lifestyle, infections. But so again, if we look at how the mitochondria divide, if it is not smooth, if we have got two DNAs, if we have got two mitochondrial DNA, They have to be one in each, but here they are divided, they kind of get jammed into one. So we have got uneven copies. Okay, again, the same players, the enzymes in the proteins involved in fusion and fission. Okay. This is what I was talking about, that when beyond a certain point, whenever there is a stress overload on the mitochondria, they release what are called as the mitokines, of which, let's say, these two, GDF15 and FGF4, they help in the communication of this mitochondrial activity whenever there is a stress overload. So this is basically what is called as the integrated stress response. Then this will decide whether the mitochondria need to be fused, fissioned, or mitophagy. Okay. So what happens? There is perturbed cellular function, there is neuronal energy perturbation, and then there is a dip in cognitive processing. This is what happens when it comes to psychiatric disorders. So if there is a regular mitochondrial energy supply, okay, that is from low energy to high normal, okay, we have got operational energy, so good cognition, which is okay, shaky cognition, cognition compromise, operational energy buffer. So these are the different kinds of things. Energy, cognition, what happens when there is a buffer? As we go with high normal, there is excessive ATP, so there is a buffer, but low normal, the buffer is lost, and so then there can be a collapse. And so this is what happens. They have tried to give a kind of a continuum picture of what happens when the brain bioenergetics suffers. So from normal consciousness, so here comes delirium, then we have kind of psychiatric disorders, common ones, and then delirium and stuff like that. Okay, for the depression, a lot of studies. So mitochondrial genetics consists of deletions, low copy numbers, variations, and downregulation of repair enzymes like polymerase gamma or POLG, which we talked about some time back. The proteome is altered. The proteins, the mitochondrial proteins are a problem. Altered levels of oxidative phosphorylating proteins. So bioenergetic problem. 20% of mitochondrial proteins are altered. 20 subunits of ETC complexes. Those 5 complexes will have subunits that are increased. Increased levels of 1 and 4 cytochrome C and ATP synthase. So 1, 4, and 5 in the dorsolateral prefrontal, anterior cingulate, and parieto-occipital cortices. Impairment in carbonic anhydrase and aldolase C in frontal cortex and cingulate. So it's not only gross, but in specific areas, there is a mitochondrial proteome alteration. So there is decreased mitochondrial ATP, decreased activity of 1+3 and 2+3 complexes, increased oxidative stress, so damage to proteins, lipids, DNA, including mitochondrial DNA, alteration of complex 1 subunits, and oxidative damage in cerebellum, and so on and so forth. Dysregulation in calcium homeostasis. More calcium starts getting in. Normally, it will not get into the mitochondrion or into the cell, but here there is a problem. And one of the candidate risks is CaCNA1C, which forms a unit of the calcium channel. Inflammation is another thing which is mitochondrial driven. So dysregulation of both innate and adaptive system, high pro-inflammatory cytokines and low anti-inflammatory cytokines, plus the mitokines. That's the depression picture when it comes to mitochondria. Mania is also similar. In the end, it is similar. The starting points are different. Starts off with increased mitochondrial biogenesis, more energy, but that drives inflammation, ROS production, okay, oxidative stress, and calcium. So this in depression, it is more of reduced biogenesis, reduced energetics, reduced DNA, damaged DNA, and things like that. Here increased, but the end result is the same. Elevated calcium, so activation of AMPK, S1RT, increased activity, pro-apoptotic pathways, increased uric acid levels, increased uptake of calcium, and so on and so forth. Now, not only that, but at animal model level, almost all psychiatric disorders have been studied and have found to be associated with mitochondrial impairments. So we talked of depression, but if you look at generalized anxiety, again, similar profile, reduced mitochondrial GTPase, mitochondrial bioenergetic pictures. Again, PTSD, bipolar, schizophrenia, substance use, ADHD, everything is associated in animal models with some or the other mitochondrial problems. Now, not only psychiatric, but let's say neuropsychiatric or systemic disorders with a psychiatric presentation. Basically, non-communicable diseases of all types are associated with some or the other mitochondrial problems. It is a very comprehensive picture, and each of these tells us a particular activity of the mitochondrion or activity related to mitochondrion which can get impaired. So, for example, if you have glycolytic activity, reduced in T2D, reduced in Alzheimer's, reduced in cardiovascular, reduced in aging, but increase in cancer. Lactate abnormally increased in all. So the metabolic map or the metabolic schemata changes. Okay. So if we look at again Alzheimer's disease, we have got mitochondrial fragmentation, which is driven by beta amyloid, activated by nitric oxide, and the DRP1, which forms the ring for fission. Reduced MFN and OPA, so reduced fusion. So mitochondria don't remain as an integral body. They split. Parkinson's disease, again, there is this pink mutation. Pink basically guides mitophagy. Now, if there is an abnormal pink mutation, more mitochondria will get damaged. And along with this MFN and OPA. So not only will mitochondria not remain intact, but they will die prematurely. Okay, this happens in cancer and so on and so forth. Then the first time anybody gets a kind of a sniff of what mitochondrial involvement is in disorders, we have this mitochondrial diseases per se. Means primary problem, only problem in the mitochondria, and they are Leigh syndrome, Alpers-Huttenlocher, MELAS, multiple endocrine lymphomas, mitochondrial ataxia, Parkinson's, and sensory organ deficits. They again have a host of mitochondrial involvement. So Alzheimer's we saw Parkinson's, multiple sclerosis, Huntington's, everywhere the mitochondria get involved. The question is where, how much, bioenergetics, ROS, fragmentation, and basically inflammatory status. This decides the nature of the disorder and where it's going to start. Amyotrophic lateral sclerosis, even migraine. So what are our treatment targets? This is the last 2 or 3 slides. So unknowingly, like all medications in psychiatry, all drugs in psychiatry, we have traditional drugs which have an effect on the mitochondria, which we have now found out. So if we look at electron transport chain, lithium valproate, oxidative stress, lithium valproate, lamotrigine, anti-apoptotic BCL2, lithium valproate, even ECT, hyperexcitable lithium, calcium BDNF lithium, lactate lithium valproate, and quetiapine degrees. So traditional drugs also have got a stabilizing or a good effect on the mitochondrial systems. So we have looked at lithium valproate, but even antidepressants, they reduce apoptosis. They increase mitochondrial biogenesis. Most of the studies are related to fluoxetine, so an SSRI. So when we continue SSRIs, it's not only that the symptoms will remain under control and SSRI thing happens. No, but at a very basic level, at a very fundamental level, it reduces cell death. So it is neuroprotective. It enhances neurotropism, reduces oxidative stress, and so on and so forth. Antipsychotics also have a similar thing. Pramipexole that we use upregulates BCL, so it is anti-apoptotic. Nutraceuticals, many of them available now, N-acetylcysteine, omega-3 fatty acids, alpha-lipoic acid, acetyl-L-carnitine, SAMe. All these have got a good effect on reducing or maintaining oxidative stress, but probably not in the doses that are kind of displayed on the back of the strip or a back of the bottle. First of all, it needs to be decided whether this is the only thing that is causing a problem, or it is a group of things in the mitochondria and in the cell itself which is causing a problem. And then we need to kind of customize. So there is no one size fits all. And a lot of other things, creatine monohydrate, leucine, isoleucine, valine, L-tryptophan, inositol, coenzyme Q, even melatonin. It increases BDNF and ERK1/2. Vitamins, ketogenic diet, folate. Now, all these last two slides from nutraceuticals and this one, they are small studies. One, they are not very kind of replicable studies. So if I have a population and I've tried one of the modalities, it may not really work in the other. So it is basically how we understand the mitochondrial system better, whether we are able to kind of really give a very basic correlation between the mitochondrial involvement and the symptoms. That is where it is probably going to work. All these things, resveratrol, taurine. I talked about that PPARG, peroxisome proliferator. That system, those agonists have got a protective value, at least in studies for Alzheimer's disease. So we have rosiglitazone, one that is used for diabetes. It activates neuronal PPAR gamma. It increases neuronal mitochondrial biogenics and bioenergetics, and it decreases the probability that mitochondrial function will fall below critical threshold where cells will suffer and they'll start getting degenerate. So this is recent studies which have shown that PPARG antagonists like pioglitazone, rosiglitazone may have a more protective role in Alzheimer's disease. Again, a lot of experimental studies on mitochondrial fission and fusion, what we can do. So different kinds of compounds, mitochondrial fusion, gene intervention, because we understand. So mitochondrial DNA, very few genes, very few proteins. We know how it gets transmitted. We can do something about it. This is what it is, the state of affairs when it comes to mitochondria with diseases. Again, if we see the diseases, Alzheimer's, Parkinson's, cardiomyopathy, so on. Not directly affecting us, but well, Alzheimer's is, Parkinson's is. Another intervention is the exercise intervention. So what happens is with exercise, the lactate utilization, the lactate accumulation and lipid utilization is better. So mitochondria become more efficient. So this is one of the studies where they have used pre-T2D patients. They have selected T2D patients in the pre-exercise, 12 months of individualized exercise program, improvements in fatty acid oxidation and lactate clearance capacity. So this is what happens. So thank you so much. And with that, I hand over to the chairperson. Thank you so much.
Thank you so much, sir, for the lucid presentation. Sir has talked about, he started with structure of mitochondria. He talked about that mitochondria has been there, has come a billion years ago, and it came from eukaryotic cells. He talked about mitochondrial DNA and nuclear DNA. He talked about that mitochondrial DNA variants is basically maternally inherited. However, the nuclear DNA has got a mixed inheritance. Then he talked about that all the metabolic things, most of the metabolism happens in mitochondria, whether it is methyl tetrahydrofolate, glutathione, urine synthesis. He talked about various types of mitotypes. He talks about metabolic map. He talked about various fission and fusion, especially DRP1 fission, and he talked about the fusion of FMN1 and 2. Then he went into the psychiatric illnesses where he talked about depression, mania, what are the various mitochondrial variants are there, what are the changes there. Then he talked about various inflammatory changes, which happens this. We talked about that brain expends about 20% of the mitochondrial and utilizes mitochondrial energy. So it has got lots of implication, not only in the finding out the pathophysiology and the pathogenesis of psychiatric disorder, but secondly, it has got an implication in the treatment part. Lots of targets are there where we can do a research and find out the interventions how already developed molecules like lithium valproate and has been there and which are affecting mitochondria. He talked about various antioxidants which have been in vogue coming, although the sample size of most of these studies have been less. So we require further research on these particular aspects, especially like lysine, and other antioxidants which are coming in the market. So thank you, sir, for the lucid presentation, and you've explained each slide so lucidly that the people who have attended have really learned about mitochondria, because it's a new topic and people don't know much about it. So I request for questions from this thing from all the audience. There is none in chat box, but I request if you have question, you can unmute yourself and ask the questions from the speaker. I've got one question, sir. In mitochondria, you talked about the cellular aging and apoptosis is this thing, which results in maybe premature kind of an aging or early aging in most of the non-communicable diseases and especially neuropsychiatric illnesses. So it is a similar kind of a thing which is also seen in leukocyte telomerase, this thing which is present in the end of the tail of the chromosome. They also works in a similar manner.
Yeah. No. So what happens is that, every cell undergoes, as I said, mitochondria are not cell specific, in the sense they are there in each and every cell. And the more active a cell is, the more number of mitochondria are going to be there. So if there is a cell in the neuron, which is very active, in the brain, which is a neuron, let's say very active, it has got 10 mitochondria in one cell. A cardiac cell, cardiac myocyte, a little bit less active, let's say we'll have 8 mitochondria in one cell. Now, what is going to happen is whenever there is a mitochondrial involvement, one is the energy that is needed for the processes is going to be allotted less. There is a change in the metabolic scheme. More of energy now or more of resources now are going to be focused on the wear and tear. So what happens is that the cell, which requires 10 mitochondria to work properly, compared to 8 to work properly, the one with 10 is going to get affected more, because slight change, the functioning gets affected. Now, this kind of a thing happens in each and every cell, and each and every cell has got a nucleus. It has got the DNA, the chromosomes, and the telomere. So each and every cell is aging. It has got its own lifespan, but this adds to that, because when critical turnover of metabolism and everything, precursors, is a problem, then even the telomeres will start becoming less active or getting more worn out easily. Because at a very basic level, it is the energy and the resources allocated. So what happens is that, now if we look at studies for all non-communicable diseases, people who suffer from severe mental illnesses, psychosis, mood disorders, on an average, they have got 15 to 20 years lesser lifespan than people who do not. So I might have diabetes or hypertension or ischemic heart disease, and somebody else who has all these 3 plus psychosis, he is going to live lesser than what I am. So any disorder or anything wrong is going to add to the telomere, because telomere is also part of the cell, which requires energy to maintain itself. That gets worn out, and now you have got more of it, so it's going to get worn out more. So yes, everything requires energy, not only energy, but a lot of these things. So Now let's say another thing, that is there is this ROS, reactive oxygen species, that needs to be cleared out. That's the oxygen O minus that gets accumulated. Now, if something is there which needs oxygen to bind to it, get oxidized, then it's fine. But by itself, it can start a problem, and the enzymes will not be able to clear it. So it's not only energetics is one thing, but all these byproducts of energy metabolism that get accumulated can also trigger by things by themselves. So a lot of things.
Okay, sir, we have got a query. Nowadays, anti-aging, antioxidant, and senolytics are buzzwords. Do they work by affecting mitochondria?
So what happens is that when there is something new, not new mitochondria are there. In fact, if they were not there, we would not be here. So it's that we have begun to understand it. And what happens is that when something like this, is picked up by lay people or lay press, then they add their own flavor to the whole thing and understanding. And okay, we already had this kind of diet in our uh, customs and society for ages, and this is what happens. We don't know exactly how those things work. And if it was so, then none of us would have, all of us would have little 150, no heart disease, no hypertension, no autoimmune changes, no, definitely no dementia and all. So we leave it at that, because even this kind of things, or the whole list which I mentioned, we can label them as anything. The thing is that are they going to work in my given patient is a big query, because this is one of the things. This is one of the things in the whole jigsaw puzzle, and it's just one piece. So even we are not able to figure out that piece as of now completely. So giving some label to something, some modality like mitochondrial modulator or something is a bit too far-fetched as of now.
Sir, we have another question, sir. Can you explain about the inheritance of mitochondrial DNA you mentioned, sir?
So what happens is mitochondrial DNA has got maternal inheritance. Why maternal inheritance? Because at the time of fertilization, one sperm and one ovum. Now they are gametes, so they have 23, 23. Now both the cells, they are both cells. Ovum is kind of stationary, so it is like a cell. So it has got the typical structure, nucleus and cytoplasm, and in the cytoplasm, mitochondria. How many per oocyte? Around 2 lakhs. That is what figures say. Sperm is a different kind of a structure. It has a nucleus which is in the head, which has got very little cytoplasm. It is actually mobile. And in fact, the mobility of the sperms, how fast they move forward, how much the tails move, is an indicator of fertility, male fertility. Now, this movement, anything that is fast requires a lot of energy. So naturally, a lot of mitochondria are going to be located in the tail area where all those myofibrils are there. You require all those protein fibrils so that the tails will move, and the sperms can move in the female genital tract. Now, once one sperm meets the ovum, the tail is lost, because the tail purpose is lost. Now only the nuclear material goes into the cell. So what happens? The mitochondria that were there in the cell, in the sperm tail, are gone. What remains is the DNA in the sperm head. These two will fuse, because you have the 23 complement from the male, 23 from the female, and form the whole set of 46, 23 cross 2 chromosomes, X, Y, XX, whatever. So what happens in a fertilized cell, just fertilized cell, we will have the full chromosome complement and maternal mitochondrial DNA, right? Now, whatever is the child, boy or girl, is going to get that component of mitochondrial DNA from the mother only. Means this crucial mitochondrial activity, which decides everything, whether it is energy, whether it is reactive oxygen species, whether it is immune modulation, all those crucial activities, most crucial being ATP, is determined by mitochondrial DNA. Now we have had something coming from the father, half complement from the father with some nuclear DNA from the father. But what it is going to determine? It is going to determine the structure of the mitochondria. It's going to determine like, okay, I'm going to live in this kind of a flat. I'm going to have white colored walls and I'm going to have this kind of sofa. But how is this going to function is maternal part. And this will go into each and every cell in different some cells are more active, more mitochondrial DNA, various haplotypes and things like that. So what happens is mitochondrial DNA, two types, one which comes half from the father, half from the mother, that determines the structure, and everything in the mitochondrium which comes from the mother, right? So this is how it is transmitted. So if in the mother there is something wrong in the mitochondrial DNA, there is some problem if we are able to figure out, then there can be an attempt to change in that ovum that mitochondria. That is what is called as mitochondrial engineering or something like that. Because we know that if it's a mitochondrial DNA involvement, it is going to come from the mother because of this mechanism. I hope that answers. Yeah.
Any more questions? If there are none, I thank Dr. Malay Dave for an excellent presentation. And he's explained everything in very simple terms. Now I hand over to Dr. Rashmi.
Thank you.
Thank you. Thank you very much, Dr. Malay Dave. Thank you very much, Dr. Vinay Chauhan. I think the brilliance of a good presentation is its complexity, yet its simplicity. And I think Malay is the epitome of that statement. So thank you very much, Malay, to tell us about the humble mitochondrion in a manner which dazzled us, at the same time intrigued us a lot. And we are beginning to realize the importance of things happening at a much more molecular level than we did earlier. So it was a poignant reminder to all of us to relook at psychiatry from the far cry of the neurotransmitter hypothesis, which we were trained in as our postgraduate student days. And this really come a long way. Thank you all for your participation, and thank you, Dr. Chauhan, for moderating this in a brilliant manner and sharing your insights as well, because it takes one to know one, right? So you were a perfect foil for Malay, and you were able to take this forward in a very, very lucid manner. So thank you very much once again for making the time and being with us. And thank you all for participating in this interesting talk this evening. And we promise to bring you more of these every month. And thank you, Sun Pharma, for your support. I wish you all a great evening ahead, and we see you again next month. Have a great day.
Thank you.
Thank you. Thank you so much, Malay.
Bye.